Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
The lens of the human eye gets its transparency and refractive power from the fact that certain proteins are densely packed in its cells. These are mainly crystallins. If this dense packing cannot be maintained, for example due to hereditary changes in the crystallins, the result is lens opacities, known as cataracts, which are the most common cause of vision loss worldwide. In order for crystallins to be packed tightly in lens fiber cells, they must be folded stably and correctly. Protein folding already begins during the biosynthesis of proteins in the ribosomes, which are large protein complexes. Ribosomes help translate the genetic code into a sequence of amino acids. In the process, ribosomes form a protective tunnel around the new amino acid chain, which takes on three-dimensional structures with different elements such as helices or folded structures immediately after the tunnel's formation. The gamma-B crystallins studied in Frankfurt and Grenoble also exhibit many bonds between two sulphur-containing amino acids, so-called disulphide bridges. The production of these disulphide bridges is not easy for the cell, since biochemical conditions prevail in the cell environment that prevent or dissolve such disulphide bridges. In the finished gamma-B crystalline protein, the disulphide bridges are therefore shielded from the outside by other parts of the protein. However, as long as the protein is in the process of formation, this is not yet possible. But because the ribosomal tunnel was considered too narrow, it was assumed, also on the basis of other studies, that the disulphide bridges of the gamma-B crystallins are formed only after the proteins have been completed. To test this assumption, the researchers from Frankfurt and Grenoble used genetically modified bacterial cells as a model system, stopped the synthesis of the gamma-B crystallins at different points in time and examined the intermediate products with mass spectrometric, nuclear magnetic resonance. The result, the disulphide bridges are already formed on the not yet finished protein during the synthesis of the amino acid chain. We were thus able to show that disulphide bridges can already form in the ribosomal tunnel, which offers sufficient space for this and shields the disulphide bridges from the cellular milieu, says Professor Harald Schwalbe from the Institute of Organic Chemistry and Chemical Biology at Goethe University. The reason for this probably lies in the optimal timing of protein production. The preliminary disulphide bridges accelerate the formation of the final disulphide bridge. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.